In the summer of 2020, I assisted Dr. Gillis in constructing a developmental cell atlas. Using single-cell sequencing data, we looked at the genes that are differentially expressed in specific cell lineages during development. I decided to focus on the cranial neural crest cell. It is important that we research how vertebrates develop to better understand a multitude of embryonic birth defects and advanced stem cell therapeutics. We asked, how does gene expression of developing cranial neural crest cells compare in frogs and zebrafish? What genes are expressed at different time points during the development of these cell types? And can single-cell sequencing help determine what genes are important for the specification of these cell types? We hypothesized that similar patterns of gene expression result in similar cell fate specification. We used single-cell transcriptomics to study gene expression at different time points of development to see similarity in the important developmental genes in developing neural crest cells. Neural crest cells are multipotent. Multipotent means that the cells can develop into multiple specialized cells. The neural crest cells develop and divide, then migrate to later become melanocytes, osteocytes, and chondrocytes. Melanocytes create pigment, osteocytes create bones, and chondrocytes create cartilage. We can see to the right, the green are the neural crest cells that are later going to become melanocytes, osteocytes, and chondrocytes as they specialize. I followed the cranial neural crest cell lineage, moving backward in time. At each stage of development, I noted which genes were expressed in xenopus and zebrafish. Next, I compared the two gene expressions. This can be seen by the pink lines. Then I followed the cranial neural crest lineage and recorded the genes by hours post-fertilization, or HPF. We also compared the data with the literature from previous research that shows similar gene expression with the single-cell sequencing data. As you can see here, the stages, I started here and went backward. And as I hovered the mouse over each point, I was able to see which genes were expressed at each stage. I also did the same for HPF. Here we can see an organized list of the genes at the different hours post-fertilization and stages. We ordered them from most expressed. The top 10 expressed are highlighted in yellow, the top 30 expressed are highlighted in green, and the top 100 expressed are highlighted in blue. Again, we collected important known genes for the development of the neural crest. These are the genes in the initial neural crest cell, and the more it develops and specializes, it becomes chondrocytes, melanocytes, and sympathetic neurons. We then organized it into a Venn diagram where we compared xenopus gene expression, zebrafish gene expression, and literature, and the pre-existing literature gene expression. We noticed that there was a lot more crossover than had been previously annotated. Moving forward, we will finish annotating the genes, and with the information we learned from our completed annotations, we will then create two Venn diagrams. One, which will compare the genes that are expressed in the neural crest in both the zebrafish and xenopus, and two, we'll compare the genes that are functional in the neural crest in both the zebrafish and xenopus. Here are the references and sources we used. Thank you very much.